Welcome, everyone. We're here today to have a presentation from Dr. Todd Ogden. He's a professor and vice chair of the Department of Biostatistics at Columbia University in the Millman School of Public Health. Dr. Ogden has interests in a wide variety of topics in both statistical methodology and various application areas. He's currently collaborating with researchers at New York State Psychiatric Institute on various statistical modeling issues with the analysis of data from brain imaging studies. Other ongoing interests include functional data analysis, non-parametric regression, wavelet methods, statistical modeling, statistical computing, and statistical education. Dr. Ogden received a PhD in statistics from Texas A&M University in 1994 under the advisement of the late Emanuel Parson. He's an elected member of the International Statistical Institute and a fellow of the American Statistical Association. He also was a recipient of the H.O. Hartley Award in 2008. He's going to give a presentation today on the analysis of shaped data with applications to mitochondria. Welcome, Dr. Ogden, and thank you for your presentation.
Oh, thank you. I appreciate the invitation, and I guess I should start by saying howdy. No one even knows what that means around here. Maybe that means I've been around New York too long, but anyway, it's great to be back in Texas, even if it is virtual, and yeah, great to see. I'm just looking at the participants. So some of my teachers and some of my classmates. It's great to have you here virtually. So yeah, what I'd like to do today is talk about a somewhat recent project I was working on that stretched me quite a bit out of my comfort zone, and so I learned a lot of really interesting stuff while working on this project, and to me it's very interesting. So I thought this might be a good thing to share with my fellow Aggies, and see if maybe some of you find it interesting as well. So my approach to this is to be pretty non-technical, and to focus more on conceptual things rather than technical things, and hopefully by the end you at least have some general idea about the approach I've taken and the conclusions I've made, and without having to worry too much about the computational or theoretical details. And yeah, I'd like to keep it informal as possible. So if you've got any questions or comments, please don't feel like you have to wait to the end. I would love to talk about anything as we go. Okay, well, I'll go ahead and get started then. So the title of my talk is Shape Analysis for Mitochondria Data, as you can see. And my inspiration for this is the third author on the list here, Anuj Srivastava, who has been interested in shape data and related things for quite a while. And he saw that I had an interest and a curiosity, and he was very kind to spend a lot of time trying to explain things to me that to him I'm sure are very simple. But so anyway, I really appreciated his time, and it led to this very interesting project. All right. So first of all, a little bit of background why we're interested in this in the first place. So within a cell, there are several subcellular structures, and in my most recent biology class was in 9th grade, so I can only understand biology at the cartoon level pretty much, but some of my biologically strong collaborators only understand statistics at the cartoon level. So it makes for a very good working relationship, I think. But within a cell, you've got to have these mitochondria to generate the energy that is needed for everything that the cell does. And in some cases, including various types of mitochondrial disease, the mitochondria become dysfunctional, and there are big problems. the cells can die, and at the least you get some gene expression that's weird, and all kinds of strange symptoms can result. But in the most severe form, mitochondria dysfunction causes it's like fast aging, and there are no treatments for this. And so it's of interest to study mitochondria, understand how they work, and how we can diagnose, how we can understand these diseases. And recently it's been found that the shape of mitochondria is important with how they function. So that's why we'd like to study some shape. So here, okay, so they took sections of muscle, and this is a mouse study here. They take very thin slices of muscle, and there's many mitochondria per cell, and they place it under a microscope and magnify it many times. You can see the scale on this 1 micrometer or 2 micrometers, and so we look at, and this is just a 2D slice of a bunch of mitochondria. And so the main question is, on the left, okay, that's a slice from a mouse that has been sedentary, okay, immediately before the mouse was sacrificed. And on the right, that's a section from a mouse that was running vigorously before the sample was taken. And there's 2 kinds of mitochondria. I can't remember what they stand for, but SS is the top one and IMF is the second one. So SS, you can see are more blobs. They're more roundish, and then the IMF have these kind of strange shapes. They're very elongated and kind of well, quite different from SS. Okay, and they have different functions. And so what we'd like to study then is there something different about the shapes for these sedentary mice as compared to the shapes of the mitochondria from the running mice? And like, how do we understand this? How do we measure this? Well, the first thing you might need to do is digitize this in some way, or some way that we can focus on shape at least. And so this was actually done, as I understand, by a very painstaking process by a postdoctoral fellow who went, slice by slice and traced the outline of all these things and made a bunch of clicks. And so the result of that is, something like this. Here we've got a bunch of different shapes, and it's, it's defined by many different points along the shape. And so here are the shapes for the sedentary, here are the shapes for the running, got both SS and IMF. Okay, so how do we compare these things? Well, so the hypothesis is that when a muscle is exercising consistently for a while, that it has an effect on the shape of the mitochondria. So this is an acute effect of the shape. And so the design is relatively simple. These are genetically identical mice. 3 of them were randomly assigned to the sedentary condition, and 3 were randomly assigned to the running condition. And from each mouse, several cells were sampled, and for each cell, many of these mitochondria were sampled and digitized, and so that it's a shape. Okay, so this is earlier work of one of my collaborators on this project. Okay, so this is a neurologist, and he originally had this data or data very similar to this, and he says, okay, I want to compare shapes. Well, how do I compare shapes? Well, I'll just make measurements on the shapes, and then each measurement, I'll just compare it from one condition to the other. I'll compare it with like a t-test. And so what are possible measurements that you can make for shape? Well, perimeter is one, surface area, circularity, aspect ratio. So these are all scalar summaries that you can measure on closed shapes like this. And I'm not familiar with most of these. And I don't know where he came up with these, but this is what he decided. He measured these things, and then he did a bunch of t-tests, and then he compared each of these across all the across both conditions and across animals and across cells. And what he found was, well, we can't really find any difference. So, you know, part of the issue is, I suppose, the one of multiple comparisons, right? You know, the more scalar descriptions you can come up with, the more hypothesis tests you're doing, and the more, you know, more likely you are to find something when there's not anything really there. But I think this is a totally reasonable approach to take. In this case, he found no differences, but I wonder that maybe such an approach could miss some information that's in the shape data, because you're only looking at a few different aspects of the shape, and presumably there are many more. And why even look at these aspects? These are not hypothesis driven. These are just, well, everything you can think of that measures a shape somehow. I think the shapes might be different, so I'll compare some summaries of shapes. Okay, so that brings me to an area of great recent interest to me, and that is what's sometimes called object-oriented data analysis, not to be confused with object-oriented programming, though there certainly are similarities. But so as we've seen, almost all of statistical data analysis involves vectors or scalars, or right? I mean, that's just sort of the way the theory has evolved and the application has evolved, and that's because that's the way that the data come to us often, right? Just in a spreadsheet. But more and more, and part of this is with new and developing technologies, we're getting data that doesn't really fit nicely into this, you know, matrix format. So for instance, so shapes, the things I was just showing, also curves and functions. That's something that I've been interested in for quite a while, right? Your basic data object is a spectrum or a time series or something like that. And then you want to compare many spectra against each other. I'm also interested in imaging. So maybe, you know, every subject you observe an image. So sure, you can represent that as a vector, but it's got some structure that I think if you ignore that, you can really miss what's going on with your data. Beyond that, graphs, that also arises in brain imaging a lot. Like these networks, you know, you want to know what part of the brain talks to what, and so you do some functional MR data, and you look at correlations, and then you draw this graph, you connect nodes, and so forth. Well, once you do that, then you've got a graph that you can do this for each subject. Then how do you compare one graph with another graph? How do you compare this across conditions or across diagnostic groups? And then going beyond that, you know, there's a lot of interest in the microbiome now, where you can represent, you know, all these, you know, many bacteria in your gut as a giant phylogenetic tree. And you can do this for many different subjects, or even, you know, different time periods within the same subject. So how do you treat that if that's your main data object? And so the idea behind object-oriented data analysis is simply to sort of unify the approach to all these things. So there's a lot that these things have in common, and so you can kind of think of you know, generalizing statistical techniques that have been developed for scalars or vectors to where they can handle more complex objects like this. And so I just found out last week, coincidentally, that Steve Marin and Ian Dryden have just published a book entitled Object-Oriented Data Analysis. And when I found out that was true, I ordered it the first day. And it's going to come later this week, and I can hardly wait. Okay, so what I'll talk about today then is one instance of object-oriented data analysis. And the object here will be shapes. Okay, good so far. Okay, so the next thing I'll talk about a little bit is the notion of shape space. And this is also a very general notion. Anytime we talk about statistical data object, whether it's scalars or vectors or functions or anything else, they have to live in some space in order to make sense of comparing these things. And so I'll focus on one particular aspect of that, and that is how do we examine the differences between two curves or between two shapes? So a shape is a planar closed curve. I say the shapes I'm looking at here today, because it starts in one point and it finishes up in the same point. But so there's a lot I can do if I can measure the distance between curves. Okay, so I have to understand what this means, and then come up with some metric that kind of makes sense with how I want to express distance between shapes. So if you think about shapes for a while and in what distance should mean is, well, for one thing, I don't think position should play into it at all. If I have a shape over here and then I have exactly the same shape over here, they're not different shapes. They're the same shape, just in different locations. So I want to take position out of it, right? I don't want that to factor into my distance. Rotation is another one. If I have a shape here and then I rotate it, it's still the same shape. So I want the distance between this shape and a rotated version to be 0. Size is another one. I'll come back to this. In some applications, you might want to rule size out. Like here's a shape. If I magnify it, it's still the same shape. Okay. In other applications, maybe size is important, and so you might want to take that into account. That's why I said maybe. And then the last one is parameterization. So in 2 dimensions, you've got an x and a y, and you can define both of these as functions of some other variable, x of t and y of t. Well, if I change the function t, I can get the same shape, but it's a different parameterization. So I would want my distance to not worry about different parameterizations. It's still the same shape. Okay, so how do you think about these things then? So shape space then can be regarded as a nonlinear manifold, and so this is a topic in topology, that a class that I never had while I was at A&M, but I really wish I had. But the idea is not so complicated. It's just you just think of some smooth surface in some space, and each shape is a point on the surface. And so if I want to say how far is this shape from that shape, then I have to think about well, how do I move along the surface from this point to that point? And so as a simple example, if you think about how far New York is from New Delhi, say, you can say, okay, well, the Earth lives in Euclidean space, and so I'll just get the the XYZ coordinates of one city and the XYZ coordinates of another, and just compute the Euclidean distance, and that's maybe sensible, but that distance will take you through the Earth, and so that maybe doesn't make sense depending on your application. And so the idea of Riemannian manifold kind of stuff is simply, okay, if I want to know the distance from New York to New Delhi, well, there's lots of places I can go, but staying on the surface of the Earth, right? I can come over this way, I can come over this way. And of all possible paths I can take, I want to take the one that stays on the surface that's the shortest. And so that's what I mean then when I say the distance between two cities on the Earth, and similarly the distance between two shapes. So the geodesic is just a path that stays on the surface of this manifold that goes from shape 1 to shape 2. And so everything in between that is also a shape. Okay, so that this is to me very fascinating in a spent a lot of time learning about it. And but I still don't understand differential geometry very well, and maybe I never will. But I am a fan, and but anyway, you don't need to understand it very deeply to appreciate what I'll talk about today, I think. Okay, but that's the idea. We're going to think about shape space, and then within shape space, we'll think about ways to measure the distance between shapes. Okay, so this is not a new problem, really. Going back to the 1970s, David Kendall defined shape as the information, the geometric information that you still have after you take out translation, rotation, and then scaling if you want to take that out or not. So that's how he defined shape. And so what he did then is he said, okay, here's a shape, whatever it is, and then put a bunch of points, discrete points along the shape, and called those landmarks. And so what that makes then is a set of m points in d dimensions. So he converted shape data to vector data. And then once you have vector data, well, you can do vector stuff on it. And so, you know, and on the one hand, it's a great way to do some, you know, reasonable analysis on shape data based on what's already been done. But there's other stuff you can do, and I'll talk about that in just a moment. But so that's certainly a natural thing to do. Just convert our shape data to vector data and then do vector stuff on it. And so there's some metrics that are based on that. Call them Kendall scaled, Kendall unscaled, and full procrustes. And I'll just show some examples of this in just a bit. But so this is one option. You know, this is sort of like old school shape space. So more recently is the idea of elastic shape space. So one disadvantage of doing the Kendall thing is you have to choose these landmarks. And in some situations, it might make a lot of sense, you know, where you would place the landmarks, right? Because it's the landmarks that you need to line up. Like you line up the landmarks with this shape with the landmarks of another shape. And you do that for all of them. And then you can, you know, measure how close they are together. Okay, but you might choose a different set of landmarks than I choose. And so we might get different answers. And, you know, even with the same shapes. So an alternative to this is just regarding each curve as a continuous function in two-dimensional space in my situation. And so if it's a continuous function, then I don't need to worry about these landmarks anymore. But I do have to worry about reparameterization, which I mentioned just a while ago. But I'm not going to spend a lot of time talking about that, except just to say that this, you know, very innovative and clever idea of the square root velocity function, it sort of handles that problem intrinsically. So anyway, the idea is you've got some shape, you got an x of t and you've got a y of t, and so t goes from 0 to something. So I'll just take those functions, and then the square root velocity, I'll take the derivative of those functions, and then, you know, take the square root of that. That's essentially all it is. And so it's a very simple transformation, and it's invertible. But the reason that it's useful, the reason it's handy, is I have this very restrictive shape space, right, with these closed curves and so forth. But when I take this transformation, then it just goes into regular L2 function space, where, you know, I personally have been interested in functional data for a long time. And so, you know, many of these functions naturally belong to L2 space. So that's great. When I get to L2 space, I have a function, you know, a nice L2 metric that I can use. And that's a perfectly reasonable way to measure the distance between shapes, right? Just transform to this other space, measure a distance there, and then transform back. Okay, so this is elastic shape space. This, I believe, should be an improvement on Kendall shape space in a lot of different ways. And similar to with Kendall, we can consider both scaled and unscaled. And you know, which one do you care about? It depends on whether size is important in your analysis or not. if you think size might, then you should use the unscaled version. And if you really want to take size out of the equation altogether, use the scaled version, which just means you scale everything to the same size and then look at the distance between the shapes. Okay, so it might be a little, I mean, I haven't even given formulas or defined these or anything, but here's kind of what it looks like. If we think about moving from a shape of a bone to the shape of a heart, there's 5 different ways I can do that depending on these shapes. Okay, so the first 2 are Kendall, and then this is that Procrustes also in Kendall space. And then the last 2 are from the elastic shape space. And so the idea is, right, so I'm traveling from bone over to heart, and I'm doing it while staying on the surface of this manifold. And so you can see, you know, all the warpings of the shape, and how long does it take me to get from bone to heart? In this case, it's, you know, 0.53. That doesn't mean anything really until you compare it from going from bone to some other shape, or some other shape to yet some other shape. So this is the scaled version. So I'm going from a bone of one size to a heart that's also the same size based on this landmark representation. Alternatively, I could go from a bone of this size to a small heart. And so it takes a different path. The shapes are pretty much the same, but you see it gets smaller as we go from here to there. So that's that other dimension that it's measuring with this distance. Okay, and then Procrustes, that's sort of similar to this thing, but let's look at the elastic ones. So, you know, again, I mean, this is sort of subjective, but to me, you know, you start out with a bone and you end up with a heart, and in here, you know, all these things kind of look like bones until they start looking like hearts, right? So it's like a really warped bone, and then, you know, around here it becomes a heart, and it comes the other way. And this is the same thing, only the unscaled version. Whereas, you know, here you got these things, you know, that I don't know, doesn't really look like a bone, doesn't really look like a heart. And so that's an argument, very subjective argument that people sometimes make in favor of these elastic shape space measures. Okay, and it has other advantages as well, but anyway, all of these things, these represent the geodesics, so the closest path between the shape of a bone and the shape of a heart, and the closest path that stays on the surface of the manifold. Okay, so we've got 5 different metrics, and the next question then is, how do we use these metrics to oh, okay. Sorry, I'm just now seeing the questions. Oh, I guess it's only. Okay, yeah. So yeah, the question is, how do you determine a heart scale to be equal to a bone scale? And so in the Kendall space, it's quite easy because you just have vectors, and so you just normalize the length of the vectors. And so yeah, it's not necessarily going to mean anything like the you know, the length or even the area, right? Those could be very different, but still be the same size. With the elastic, it's yeah, I can't answer that question well for the elastic. I'd have to look into that, but for the Kendall, it's clear. But okay. Okay, so these are the distances then. So once we get the distances, what can we do? And so here's where we have to think about extending stuff we know how to do on vectors to, you know, stuff with shapes or even more abstract objects. So I'll say, okay, well, here's the very basic problem, is I've got a bunch of things that come from some distribution function f. Then I have another set of things that come from some other distribution function. Oh, hey, Val Johnson has a question. Sure.
Hey, Todd. I was just curious. So the mitochondria are measured on a 2D image, but they're actually three-dimensional objects. So does this extend if you're doing the projections of the three-dimensional objects into 2D?
Yeah, so it's not, yeah, you can't really think of it as a projection. It's really just a slice. And so there's a lot of randomness to which slice you're going to get. Because they're like, how you happen to take it could really influence these things a lot. If you have a big enough sample, then you sort of hope all that stuff balances out. But right, all you can really claim then is based on the slice, and there's going to be some randomness in choosing that slice. So the better analysis would be based on the 3D shapes, and I'll touch on that at the very end.
Okay, thanks.
Here. Sorry, I'll try to pay better attention. But yeah, thanks for the questions. I appreciate that. Okay, so here's a very fundamental idea here. I've got two random samples from two different distribution functions, and I want to compare them. Okay, well, let's suppose that I have a distance measure. Okay, so I got to have that. And so what do I have here? Well, so I think this is a reasonable way to test for a difference between two distributions, a random sample from each of two different distributions. So this first piece here, I'm taking the distance from everything in the first sample to everything in the second sample, right? So it's all possible combinations here. And then down here, I'm taking the distance of everything in the first sample with all the other things in the first sample. There's some redundancy there. And then over here, I'm doing the same thing with the second sample. So the distance from all the bees to all the other bees. And so this maybe looks somewhat familiar. This is like sum of distances between two populations. And then this is like sum of distances within a population and another sum of distances within a sample. So, I mean, this is the basic idea behind, one-way analysis of variance. You compare between sum of squares within sum of squares. Now, when you're doing analysis of variance, these are just Euclidean distances, and you get with some distributional assumptions, you're able to work out what the rejection region ought to be and so forth. But the idea is the same, even though it might be a little bit harder to figure out things like what's a p-value based on this sum. And so we can simplify notation just a bit by saying, okay, here's this g function. I provide two sets to the g function, and what I get back is the average distance of all the pairwise distances from the first set to the second set. And so this so-called energy statistic can be written like this. Okay, so then maybe it's more clear here, right? This is the average distance between A and B, the average distance within A, and the average distance within B. And so, it might be hard to figure out p-values, but you can always do things with permutation testing, certainly with a simple design like this. Okay, so that's great then. All I need is a distance and some samples, and off I go, and I can do this energy statistic and test for differences. Okay, and that's comparing two groups. If I want to compare k groups, it's really kind of the same idea. I've got k different distributions, and I get a sample of shapes from each one of those distributions. It doesn't have to be shapes here; it could be any object. And then the same idea. I'll get the within sample. So comparing all the shapes within sample AI with all the other shapes within that sample. And then the between sample is everything from AI to AJ, where I is not not equal to J. So I put it all together, and the same idea. I've got a within sum of squares and a between sum of squares. And if I'm still thinking ANOVA, I could take this ratio and off I go. And this is another thing that I can solve with a permutation test in most situations. Okay, so that's great. You know, if only it were that that simple. With this particular design, though, it's like, well, what if the samples aren't really independent? Okay, so it would be great if I had, you know, a set of mitochondria from one condition that I can regard as a random sample, and then another set of mitochondria from another condition that are a random sample. Then I'd be done. But the design's not quite that simple. What I have is within each condition, I've got several animals. Well, several means 3. And within each animal, I've got a certain number of cells taken. And then within each cell, I've got several mitochondria. So this is sort of the design of the study. Animal 1, from the sedentary condition, 5 cells were selected, and there were 20, 41 mitochondria per cell. Anyway, so these numbers are, you know, somewhat consistent across animals and across conditions, but there's some structure here that I don't think I should probably just ignore. So, you know, I don't know, like what's, you know, what is, if I were just thinking in terms of ANOVA, this would be, you know, no problem. I'd say, okay, well, cell seems like a random effect. So I'll model that as a random effect. And animal also seems like a random effect. So I've just got this hierarchical model with one fixed effect, that's the condition, and 2 levels of random effects, and off I go. But with this distance-based statistic, it's not quite so simple, at least not to me, how you would do that. And I haven't been able to find, you know, in any work that's been done on you know, test statistics for things like that with arbitrary distances. And so the first question is like, well, you know, maybe the cells are not really, you know, that different from each other. And so, right, if all the cells are kind of the same as all the other cells, then maybe I don't need to worry about cell as a random effect. And so, well, you can test that, and there's probably other ways to do this, but all we did here is he said, okay, well, within each animal, there's several different cells. And so I'll compare each of the cells with all the other cells. And by doing that, I mean, I'm comparing the mitochondria within a cell to the distance between the mitochondria between cells. And so I'll do that test statistic separately for each animal. And, you know, since I've got these different metrics, I can do this again for each metric. Okay, but I did it separately for each animal. So animal 1, okay, well, the ones I want to focus on the most are these elastic ones. I think these are the better metrics, and the other one, the Kendall ones, are just sort of here for comparison. But the scaled version, so that's where I don't care about scale. If they're big, if they're small, I don't care. I'm only interested in the shape. Well, for mouse 1 here, they're very clearly different from one cell to another. For mouse 2, mouse 3, they're not so different, right? They look like they're almost, you know, exchangeable. The unscaled, I get kind of the same story, right? They're clearly different for animal 1. Animal 2, they're probably not different. But animal 3, they're clearly different. So I think what this is maybe indicating, right? I see this in the unscaled, and I see something quite different in the scaled, is maybe the average shape size is quite different from one cell to another within animal 3, even though the shape itself is not so different, right? Because this unscaled thing is kind of a combination of shape and size. And anyway, so we do that for the different types, the 2 different types of mitochondria and the 2 different conditions. And, you know, what do we conclude from this? Well, we said, well, you know, it would be great if we could ignore cell. I just don't think that's justified in this case. For some animals, you know, it might be okay, but clearly not for all animals. So, so yeah, we're going to have to think about this nested structure somewhat. So, yeah, okay, I'm sorry. So this, I guess, is out of order then. I should have talked about this before. But yeah, so here is the distance between cells. So that's the next thing we could do then, is say, okay, well, instead of a mitochondria as, you know, the unit, maybe I should have the unit be a cell that is a collection of many mitochondria. And so how would I compare one cell with another cell, whether it's in the same animal or whether it's between animals, between conditions, or whatever? Well, so I'll just apply that statistic I had before, which is, right, so this G is, I'll take all, you know, the within differences between all these cells, and then the, yeah, that's between, and then this is within cell. Okay, and so now what I have, so I started with a distance between shapes, and then, but based on that, now I've got a distance between cells, which you can think of as a distance between average shapes within a cell. Okay, so now this gives me a tool, and again, it might not be a very sharp tool. It might be kind of a blunt tool, right? It's not quite as elegant as, you know, like having a random effect of cell in there, but still, it's something. I mean, I think it'll give us valid inference. And so based on this, now I can compare cells, and so I'll do that now. So I'll say, okay, now I'd like to compare the animals. So I haven't yet compared conditions. I just want to compare animals first of all. And so again, here are all my metrics. And what do I find? Well, okay, the sedentary mice with the SS type of mitochondria, if I do this test, then for all of these metrics, I don't get anything, you know, even close to significant. And then a similar story here with the sedentary, the other type of mitochondria. Similar story again with the running condition, but with the running and IMF, these things are quite different. And so, you know, the IMF, those are the sort of long elongated mitochondria, where the SS ones are more blob-like. And so, you know, that's kind of unusual. So we went and looked at the individual animals within that condition and in this particular mitochondria type. And what we found is, so we compared, you know, animals 2 at a time. So 4 versus 5, no clear difference in any of the metrics. Animal 5 versus 6, no clear differences between any of these. But 4 and 6 seem quite different from each other. And you know, this is the kind of thing that statisticians, you know, have to be able to explain, right? So we say 4 equals 5 and 5 equals 6. So therefore 4 equals 6, right? Well, no, that doesn't hold. So anyway, I think we all understand this from our excellent training from Texas A&M in analysis of variance. But so that's what's going on. It looks, well, maybe that's what's going on. So it looks like the difference between cells is relatively large. But the difference between animals, for the most part, is relatively small. And maybe that's not so surprising because these animals are genetically identical. And so, I mean, this, when I explained this to my, you know, neurologist friend, he said, oh yeah, well, that's kind of what I'd expect, right? There's a lot of variability one cell to another within an animal, but between animals, there's not much variability in cells. And so that's sort of backed up by this. Okay, so I've got my 2 layers of random effects, cell and then animal, and I cannot ignore cell. I mean, I have to take that into account in some way, but maybe it's justified to neglect the random effect of animal, because whatever variation that is is small, with the possible exception of this IMF. Yeah, I think we're more interested in the SS anyway. Okay, so finally, then, we're able to compare. So there's, I can compare the two different types of mitochondria, SS with IMF, and then I can compare the two different conditions. So comparing mitotype is maybe not so interesting, right? We compare the SS versus IMF within the sedentary condition, and they're clearly different. And that's just no surprise, when you look at those shapes, and then do the same thing for the running condition. But really what this is all about is comparing conditions. Okay, so here's what we get. So the SS is probably what we're more interested in, sedentary versus run. So I'm treating animals as exchangeable, but I'm measuring distances between cells. And a distance between cells mean just, averages of all the pairwise distances of the mitochondria within the cell. Okay, so the scaled versions of these things, so the scaled Kendall and then the full Procrustes, give me something around 0.01. The elastic gives me something that's slightly below the magic number 0.05. The unscaled, there's no clear difference. And then in the IMF, there's no clear difference in any of these things. And so, what would I conclude from this? So, I don't want to make too much of this 0.04, right, with all the studies that we did. But if I were to interpret this, and I'd say, okay, and this is the scaled difference. So we're ignoring scale, only looking at shape. So because if I include shape in there, then whether I'm doing Kendall or elastic, it doesn't look like there's any difference at all. But maybe I can bring myself to believe that there is some difference here with the scaled versions, so that the shapes are maybe somewhat different between these two conditions. Again, it's not very strong, but it would make sense, right? That the shapes I don't care about really, but the sizes I don't care about, but it's the shapes I really care about. And so when I focus on the shapes, like I'm doing on the scale, I might find some difference if it's really there. Whereas here it's getting mixed up with the size that maybe I shouldn't care about. So that's sort of where the analysis ends. Then I've got a few discussion points here that are pretty random, and then I'll be done. So a question that I'd really love to have the answer for is, well, how do you handle object data with a hierarchical structure? What I did, right? I mean, is I've got my metric, and maybe I believe in that metric, comparing one shape with another shape. But when I get collections of shapes, all I did is I took the average, between and within. And so, that's fine, it's okay, but maybe it's not not optimal for anything. And, give me some reasonable answer, I suppose. But there's got to be a better, more principled way to do this, which I haven't seen, and I don't know. And then some comments more on the application. So yeah, the SS mitochondria are more circular, right? They kind of look like blobs, elongated blobs a bit, and so their shapes are not so wildly different as compared to the IMF, which are these long skinny strands that some have branches out and so forth. So I take this, so I say changes in their shapes may be easier to detect. And what I mean by this in like vector terms is I think the variance, right, like the noise variance in SS mitochondria is smaller in some sense than the noise variance in the IMF. And so in that case, a smaller true difference is going to have more power to be detected, and all this by analogy. And I mentioned this before. If you have the scale invariant shape matrix, then it allows you to completely ignore the size of these things. And so if there are, small shape differences, then, it's sort of like taking out one source of variability, the variability in size. And so it gives me more power to find, even more subtle differences in shapes. Let's see, elastic metrics are better than Kendall. I think that's true. I think there's certainly more power there. And then to Val's point, yeah, so mitochondria are really three-dimensional, and so that is a major limitation of what I've done here. And I said it wasn't a projection, but here I say it is. So it's not like I'm taking a three-dimensional object and projecting it onto 2D. It's simply a slice, and a random slice at that. And then I'd also point out, I've been treating the mitochondria within a cell as a random sample, and that's probably not exactly biologically correct, because it's known, and this is fairly new and exciting stuff too, that mitochondria within a cell interact with each other, and possibly even between cells. So I've seen some videos of mitochondria in one cell and another cell close that seem to interact with each other. It's really kind of freaky. It would be really fun to measure that and to understand it better. But that's all I've got. Here are a few references, and I thank you for being here. And if anybody's got any comments or questions, I'd love to chat about those.
Any questions from anyone in the audience?
It's Di Michelson.
Hi,
How are you?
Real good.
So this is fascinating to me, and I enjoy, not to the point of research, but I enjoy functional data analysis, and I teach now, and so I teach that as part of my repertoire. So I'm wondering, in functional data analysis, of course, you have basis functions that represent archetypes of the shape of the function. And I'm wondering if maybe it's already built in here, but are there, archetypical shapes in 2 dimensions that then you can build off these basis, I don't know what you'd call them, right? Basis shapes. And is that what's happening in these distance-based statistics or not?
That's a great comment, Di. Yeah. So as soon as you define a metric like this, then you automatically get a mean, right? So the mean is the shape that minimizes the squared distance to all the different shapes. And so yeah, you're able to get a mean of a data set, and the mean is a shape. And then you can start thinking about things like principal components. And so now principal components is going to be shapes. It's a little bit harder to represent, but I guess you've seen it with functional data where, you got the mean function, and then you got all the little plus signs, if you're up one principal component, and the minus signs if you're down one. And you can do similar things with shapes, right? So the first direction of most variation is, and this is what it looks like if you're mean plus 1, and this is what it looks like if you're mean minus 1. As far as like representing shapes in terms of basis functions, I'm not really sure. You can certainly do that for computational, but I'm not really sure if that idea extends well to shapes.
All right, cool. Yeah, thanks. That makes sense.
Thanks, Di.
Todd, this is Kathy.
Sure. Hi, Kathy.
Hi. Just great presentation and great work. Super exciting, actually. I was wanting to go back to the idea that you're dependent on the quality of the slice of the mitochondria. Is there any, do you have concern there that there might be lab issues that maybe you shouldn't, that's a big issue that needs to be accounted for that is not? Or you think that it just gets rolled up into the sort of the noise of the system?
Well, I think what I'd worry about would be bias if there's some particular direction that gives you like a different idea, you know, of like what the shapes look like, than if you were to do it in some other direction, then that would certainly be a concern. I haven't had any conversations about this, but my impression is just like, yeah, you know, a muscle is a muscle and we slice it, and it doesn't really matter which way we slice it. But that may not be true. I've never asked that question.
Okay, thank you.
Hi, this is David Allison from Indiana University on cell phones. Really great presentation. Thank you so much. One of the things I'm interested in is the idea of rigor and reproducibility and replicability of analyses, and also detection of p-hacking. We think about this especially in epidemiologic work, where there's so many different model choices one could make. And I'm wondering, I've been thinking lately about considering the statistical model fitted to the data as an object, and then trying to create distance functions that characterize the relationships among the different statistical models that are fit or could have been fit, and then compare result similarity to model similarity with those distance functions. And I'm wondering if you have any thoughts on how to calculate a distance function that would make sense, treating a statistical model itself as an object.
Yeah, it makes a lot of sense. I've never thought about that myself, but I guess that's kind of what Bayesian folks do when they do like model averaging and so forth. So that must induce some kind of metric, and that would be like, you know, distance between two models. Or I don't know, meta-analysis. I'm just thinking out loud here, but in meta-analysis, I guess you kind of do that when you want to. Well, I guess that's maybe the same model but different data sets. But yeah, so different models crossed by different data sets and distances. Yeah, that makes a lot of sense. I haven't really thought about it, but I think it's a great idea.
Great. Well, maybe we can get in touch and think about it together. Thanks for a great presentation.
Oh, thank you.
Dr. Ogden, there was one question in the chat from Taylor Petty.
I'm sorry, I missed that.
I took Steve Marin's OODA class, and he did a lot of PCA on various forms. Have you done PCA on this shape data?
No, I have not. I'm very envious that you took Steve Marin's class. I wish I could have taken it, and I'll get the book soon. So, no, I have not done PCA analysis on the shape data. I'd like to, but I haven't yet.
We still have a couple more minutes if anybody has a question or a comment. Seems like there's not any more questions or comments, Dr. Ogden. Thank you very much for an excellent presentation. It's a very exciting topic for many folks. Appreciate you sharing with your fellow Aggies.
Well, thank you. I guess I should say gig'em. Yeah, thanks for coming out. It's great to see you folks.
Thanks, everyone. Have a great day.